Okay, hi everybody. I think we'll get started. It's lovely to see such a full room, and I'm really glad we're somewhere where we can see the sunshine, which is nice. So I'm Ali McIntosh. I am the director of SALSA. I'm delighted to be here today. I'm working with colleagues at the Data Lab. Thank you so much all for coming today, and a warm welcome to everybody online as well. So just in terms of, there's no fire drill planned. So if there is a fire drill, please do go outside the front door and wait outside in the lovely sunshine. So today is a session all about the intersection between life sciences and data sciences. And so I'm very much from the life sciences background, life sciences being everything that is living from animals, humans, plants, microbes, so across the whole spectrum. And within the discipline of life sciences, data is really crucial and has great potential to be able to unlock lots of exciting, wonderful things and have great societal benefits. And we're going to hear about some of those things today from our speakers. So the way that things are going to work today is we have got three fantastic speakers, two researchers from universities here in Scotland, and then Hugh Wallace is here with us from Research Data Scotland. So I'm going to get my clicker in 2 seconds. And I'm just going to take you through a very brief overview of who the organisers are today, who we are. So this event has been organised as part of the Data Lab community. It's a community of over 6,000 data and AI professionals, students and enthusiasts, some of you who are here today. And they run regular events and webinars and find like-minded peers, ask questions and gain support all on the community. So you can join via the QR code here, and it's free and a really exciting platform to be part of. I'm here today from SALSA. So SALSA is the Scottish Universities Life Sciences Alliance. We are an alliance across 13 higher education institutions in Scotland. At a very basic level, we're all about driving collaborative life sciences research and supporting life sciences research here in Scotland, working both nationally and internationally. So please do today tag us on social media. You'll find us on LinkedIn, both SALSA and also the Data Lab, and also on Blue Sky as well. So with that in mind, checking my checklist, I think I have said everything I wanted to say. And I'm going to hand over now to Nick Rattray, who's here with us from the University of Strathclyde. Thank you very much, Nick.
Thank you very much, Alan.
You can. There we go.
Cheers. Hi, everybody. Thanks ever so much to the Data Lab team and the team at SALSA for the kind invite to talk to you a little bit today about the work that I do and my research group do at the University of Strathclyde. So I am not really a data scientist, I'm sorry. I'm very much more of a clinical biochemist. But what we do is we try to understand biological models of aging and try to use different analytical tools to measure chemistry within those models, and then use data and AI to try to work out things like mechanisms of disease, mechanisms of aging, biomarkers, and potential druggable targets. And what I'm going to talk to you to do a little bit today to start with is, I must apologise, you are going to get a little bit of a lecture about biochemistry within this, okay? So this first slide is, in order to measure aging in a biomolecular or biochemical model, first of all, we've got to look at the way in which sort of biochemistry links together. And most of us will be aware of the central dogma of molecular biology. In your cells, you've got a nucleus. In there, you've got chromosomes. They've got DNA. And that basically allows us to give the blueprint for the proteins, ultimately those guys there, the point doesn't work, but the proteome, which is the proteins within our body, which give us structure, but also gives us ways in which we can build down and break down stuff within our body, like sugar, to give us energy. So that's how we can kind of understand biochemistry. And ultimately, that's the chemistry of life. Okay, all of that stuff changes as we get older. But within aging, there's another framework within which we can hang all of that chemistry onto. And it's this thing called the hallmarks of aging. So there's 12 things that scientists have kind of identified within all of us that change as we get older. Now, we can probably relate to things like genomic instability, as we'll be aware that our genes change, our genetics change as we get older. Things like senescent cells. So cellular senescence is, as I've got older, I've got more wrinkles here, right? That's my collagen cells that have turned senescent adolescent. It's cells that are not really doing what they should be doing. And ultimately, what we need to do to understand aging, and actually, it's not just aging I'm keen to understand, I'm actually keen to understand frailty, and this concept of a clinician can look at a frail individual and go, that individual looks frail on a scale of 1 to 10. But biomolecularly, with all this stuff, we don't understand frailty at all. So I'm really keen, I'll give you a couple of examples shortly, I'm really keen in trying to operationalize all of this. I'm sure you can understand, by trying to understand all of this, the data that we generate in our analytical models that we get from it is pretty big. So the way that we measure that data, there's two main ways we measure it. One is through sequencing arrays, that's our genome. Another one is through this mass spec and NMR, and that's what I do in my lab. That's the main technologies that we do. Ostensively, for those that are not familiar with any of this, what you get out of this is a spreadsheet, right? You get an XML spreadsheet, a text file, or whatever it is, data array where you get a random or name of individual or pseudonym of individual, and then you get a list of the chemistry that you've measured there, and then you get the data array in the middle. So you get an expression value of all of these millions of SNPs, hundreds of thousands of transcripts, tens of thousands of proteins, and probably thousands of metabolites as well. To give you an idea of how complex this data is, in metabolomics, and I'm sure you've all been on the human metabolome database, it's a classic, so it is, and you've basically, the scientists across the world have measured about 250,000 metabolites in people. That's 400 of them, and how they relate together. That's just the metabolites. That's not the proteins, the transcripts, and the genome on top of it. And we have to understand all of that to really understand aging and frailty. Hence where data and AI and all of this stuff is going to be really important in our area. So I've got two quick projects for you. One of them, this legend down in the left-hand corner, is Jean. Jean is an ST7. She's going to become a consultant gastroenterological surgeon. She basically has performed this clinical trial Jean sat in my lab. She's performed this clinical trial. We've taken 150 individuals that have had emergency laparoscopic surgery, and that is individuals that are over 65, and basically they've had a 10-inch scar put down here, and they've operated on their colon. And if you're over 65, clinically classed as being frail, 23 or 15% of those 23 in this trial, out of the 150, died before 90 days. And that's because they didn't survive the stress of the surgery. This is where frailty comes in. These individuals had an underwritten sort of chemistry within them that we couldn't identify, that basically said they weren't going to survive that surgical process. So we're very keen to use the tools, those mass spec and all those analytical tools that I showed before, to try and understand this process further. So some of the ways we can do it is we've analysed about 1,000 blood samples using one of the technologies that we've got, and we measured about 1,000 metabolites. Statistically wise, what we've done is we've done some univariate stats initially on those metabolites to try and work out, I suppose initially, before and after surgery, does the metabolism change? Out of those 1,000 metabolites, we did this thing called a marginal means HCA. Marginal means because it means across the samples, and HCA means it allows us to show the statistical clustering between the different metabolites that we've got. And I'm going to focus in on this little group here, this group A here. Basically, 130 of them came out statistically significant within the model, and the HCA basically means they cluster together if they're similar. And I'm just going to skip through to this point here. So this is just those 11 metabolites, and we took 5 blood samples from a patient. This is pre-op, post-op, 7, 30, 90 days. And what you can see is with those 11 metabolites that we measured from their blood, if we get to post-op, and then this yellow line, this is 11 individuals that were identified as non-frail before surgery, but sadly died at 30 days. If we took 11 of them, the value of those 11 metabolites quadrupled over that 30-day period. So instantly you're thinking that could be a really useful biomarker panel. If clinicians got that before they had surgery or just after, they could track that and then maybe put those patients into some form of enhanced care that they could get from the back of it. One of the other things that we've done as well, we do things like lasso regression and regularized statistics on this as well. And the idea behind this was to try and take that blood sample before surgery and try and then predict to see if there's anything in that sample before that could actually show that the individual isn't going to survive it. Not to go into too much detail, lasso allows us to basically do a linear regression and then shrink all the coefficients down to 0 or things that are significant in the model. We did that, and the kicker from this is we had about 15 metabolites. If we take the age of the person and 15 metabolites in the model, 96 times out of 100, we can predict if that person is going to become more frail or die before they have the surgery. So I'm obviously very interested in this. We've got a validation cohort that we've actually just finished analysing in the lab. If it validates, fingers crossed, it would be actually quite a big thing. It'd be a really, really big thing. So that's one of the ways in which we use big data in frailty. The other way in which we use it is, Lisa is one of my PhD students in my group, and we're very keen to use the UK Biobank as a resource as well for looking at data. I think 500,000 people, and we can do all sorts of things with that data. It's got full gene sequencing. It's got 50,000 people that have got 3,000 proteins looked at, and that's what I'm going to talk about here. What we've done in the UK Biobank on those 50,000 individuals, we've developed a thing called a frailty index. These people are not frail, these people are frail. And it gives us a mechanism to be able to then map all of that sort of omics data over that distribution to then say, right, what proteins and what metabolites, what other chemistry is different between those ends. So we've done that. This is the walkthrough in which you've got 500,000 people of UK Biobank participants. We've got some that have got proteomics data, 53,000, and then we do all sorts of missing data imputation, adjust for covariates using age and sex and stuff like that. Then we did some regression modeling. So we did 7 different regression models, again, to be able to try and predict which of these proteins are good at separating out frail and non-frail individuals in this huge cohort that we've got. We did the standard hyperparameter tuning. Please don't ask me questions on that later on, within the stats that we did. And then what you do is you get a ranked list of proteins at the end of it. Like, we did the gradient boosting, which basically, again, allows us to try work out if the model's good or not. And then we did this thing called a SHAP, basically generating SHAP values from this regression, which allows us to select these different proteins. Out of 3,000, we've got about 25 different proteins that we now think are far more expressed in those individuals that are frail in the UK Biobank than are not frail. Now, when we get this data, what my job actually is not just to measure all this stuff, it's to translate it. It's to go from measured chemistry into how does that change a cell? How does that cell then change tissue? How does that tissue reflect onto an organ and then onto a person as well? So one of the next stages that we have for doing that translational step is that we pathway map it. So there's that map that I showed of the 500 metabolites, we actually know what that map's like for the 250,000 of them. So we then begin to place those proteins and metabolites onto that map, and that allows us to be able to then, again, do that translational step. So we can see from this experiment that took us about 2 months in the UK Biobank that those frail individuals there seem to be links to different hormone activity, different aspects of feeding and different aspects of blood circulation. And again, we do more experiments then in the lab. We might take cell models and other different types of models to try to validate this. So in summary, a lot of the work I try to do is to develop biomarkers and develop ways in which we can detect disease quicker and intervene with it. And 70% of all medical diagnoses are dependent on the lab diagnostics that we generate. In the US, it says there 7 billion clinical biochemistry tests are performed in a year just in the States alone. And I think it's actually worth more than $500 billion now globally because of it. And the thing is age is the far biggest confounder in just about every single disease that is out there. If you live to 100, you've got something like a 70% chance of getting cancer. It's just because of the statistics of it. So 51% of total disease burden is related to age. So as a final question, if we've got $500 billion worth of clinical biomarkers that are getting spent every year, what's the global value of clinical biomarkers for aging or frailty? Has anybody got an idea? The answer is 0, because there aren't any. And on that, I'm finished. Thank you very much for your time, all right?
Thank you so much, Nick. That's fantastic. So it's my pleasure now and if you do have questions, keep them in your head, or write them down, and please do ask the panelists later. And I should use my microphone. Apologies for that. So just in online as well, if you have any questions, please do put them in the chat, and we'll be able to put them to the panelists later on. So it's my pleasure to introduce Caroline Franco, who's here with us from the University of Aberdeen and the Institute of Applied Health Sciences. And she's going to speak to us today. Caroline, I'm going to ask if you're okay to stand there for me so I can see your home. Thank you so much.
Yeah. So thanks everyone for being here in this sunny afternoon. Thank you so much for the organization committee for inviting us. I'll talk a little bit about something slightly different. So I'll talk a little bit about how we use data, particularly public health related and data in mathematical modeling. Just for context, my background also a little bit different. I come from a physics background, but always interested in interdisciplinary, multidisciplinary applications of that sort of framework and mathematical tools. I started interested in more basic biology and population dynamics sort of things, but quickly my interest grew towards applications in health sciences, healthcare, global health. So Now I am in the School of Medicine in the Institute of Applied Health Sciences in Aberdeen. And I'll talk a little bit about the modeling work that I've done recently. I'm also the lead for the Masters in Health Data Science there, where we teach some data science in general, but also very much applied to health and life sciences problems. In terms of research, I'll focus on one example in the interest of time, the experience that we had during COVID. Everyone is sort of tired of hearing about COVID, but I think it's very relevant in the sense of being a paradigmatic moment, not only in terms of global health, but also in terms of understanding and use of mathematical models to inform public health. We saw many types of models. We heard a lot about the R number, the predictions, and all this sort of things. Where is the peak going to happen? And I have a lot to say about these things, but maybe we can talk about it later. But I'll talk a little bit about my experience as part of a couple of consortia trying to voluntarily respond and support public health organizations. So I was part of the Observatorio, which was a consortium in Brazil that started only with our research group and then later on expanded to a national level initiative with collaborators in all states of Brazil. We basically produced data analysis, models, dashboards for in different cities, in different states to keep track of the stage of the pandemics and also produce estimates and compare intervention scenarios, that sort of things. And I was part of the COMO, or COVID International Consortium. That is something that started in the University of Oxford, but then we started expanding to other collaborators across the globe. And we developed a mathematical model, which is a mechanistic dynamic model, with a lot of expert input, where we were able to apply that model for different settings, fit it to different countries, different intervention strategies, and so on, to compare and help policymakers make decisions in terms of what strategy is the best. So that's the sort of stuff that we did. We collaborated a little bit with WHO and different countries' ministers of health to try to give a little bit of information, particularly in low and middle income countries. So these are a few examples of what this kind of model can do. So mathematical models are quite different from the general things that you think about when you think about AI or machine learning. They are not data-driven. They are more based on expert knowledge. So we write a model based on the mechanisms and the things that we know so far about the disease. And they are very relevant in the context of comparing the impact of different pharmaceutical or non-pharmaceutical interventions on a population. So particularly these examples are population level models. So for COVID, even though it's a single disease, we developed very different types of models to answer different questions related to impact on non-pharmaceutical interventions, such as lockdown, social distancing, models to estimate a particular parameter, transmissibility, when we started detecting new variants, models to evaluate the impact of different vaccination strategies, different school reopening strategies, and also more recently, or actually I think we finished 1 year ago, a collaboration commissioned by the UK Health Security Agency to develop sort of a modeling tool to help design testing strategies. So we were evaluating what happened in the past and also trying to develop a tool for the future. So in case there is, for example, a new pandemic and you want to develop a better optimized testing strategies. So that's the sort of stuff that you can do with this type of model. And what is different from data-driven AI, other types of models, is that maybe you don't have enough data, maybe you don't know everything. There are a lot of unknowns, especially with any emergent diseases. So you need a little bit more in terms of expert knowledge to make any predictions. You don't have any past patterns to prevent the future, right? So you cannot simply train a model based on data. And that's the kind of output that we can have. We can calculate our numbers, which is something that sort of tells you how fast the disease is spreading. You can compare different interventions, so different lines or different interventions. You can, make this a sort of optimization or sensitivity analysis for the different exploring the parameter space for different coverages of different interventions. So that's the sort of thing that we can do and that we did in this context. Those projects happened and finished a couple of years ago. They are still very relevant in terms of the framework that we develop. I'm still working on some of the learnings, like for things related to heterogeneity in the populations, the things that are very difficult to consider in those models and that we learned that are so important and they affect the outcomes at population level so much. So that's one of my current projects. But just to wrap up this quick talk, I would like to also blow the whistle a little bit about our Aberdeen Center for Health Data Science. I do mostly this mathematical modeling type of work with applications in public health, global health, not only COVID, but different infectious diseases. But Aberdeen Center for Health Data Science, which is actually being rebranded as a Biostatistics and Health Data Science group, has a lot more to offer. There are like different projects going on in terms of applications of AI data science modeling in healthcare, global health. We have also different training opportunities available. I myself coordinate a couple of master's programs and courses, but there's a bunch of options there. Some of these big projects, not necessarily I was related to, but for example, the use of AI to detect cancer, or there was the verbal autopsy project where you use basically natural language to try to understand mortality in some low and middle income countries. These are all the kinds of things happening in the center, and it's all data science and modeling in the context of health and care. I think, that's what I wanted to say in this short space of time that we have. If you have any questions later about modeling, you can come to me. Also, feel free to take a look at the current research projects that we have in our website or contact us directly. Thank you.
Thank you very much. Thank you. So our last and final speaker is Hugh Wallace, who's here with us as make sure we get it right, Chief Information Officer at Data Research Scotland. So it's lovely to have you. So I will pass you on the clicker, and there we go. Thank you.
Thank you, Ali, and hello, everybody. I've been asked to stand at this side, so there's no way disguising the fact that I have got notes to read off bits and pieces. But I mean, thank you first to Nick and Caroline for some really valuable and in-depth perspectives. I should probably move the slides on, shouldn't I? Here we go. So really valuable in-depth perspectives. What I'm going to talk about goes a little bit broader, and I'm hoping will still be of interest to you all. First though, some art. When I'm asked to talk about AI, I quite like to remind people that art created by human beings is still important. And I really like the sentiment of this piece. It's by Nathan Coley, and it is on display in Edinburgh at the National Gallery of Modern Art. AI promises us miracles, new ones nearly every single week. But maybe there are no miracles here, just new tools and new processes and new thinking to absorb. As Ali says, I'm Hugh Wallace. I'm the Chief Information Officer at Research Data Scotland, and our organisation exists to make it faster and simpler to access Scotland's public sector data for scientific research purposes. We're a charity, and we're based in Edinburgh. The current picture, our problem statement, why our organisation exists really, is that access to data is somewhat fragmented. It's spread across lots of different organisational silos. And we're in the process now of consolidating processes and working out how we can streamline procedures. We launched a researcher access service a year ago, which means that researchers can, for the first time, get access to data via an end-to-end digital process. I should point out that this isn't open data, it's sensitive case-level data held in a trusted research environment. And in the fullness of time, there are 9 data sets at the moment. We want to be able to offer data that can link different disciplines and hopefully achieve different outcomes. So to take Nick's example, if we can give researchers the ability to access data about ageing and frailty with data about health outcomes, with census data, you can start to pinpoint, you can start to think about longer term solutions and longer term interventions. These are the kind of core principles by which our organisation operates by. I won't go through all of them, but the first one is pretty fundamental. A public good assessment is a key part of the data application process. And as we'll talk about over the coming slides, public good and artificial intelligence aren't always natural bedfellows. So to get into the subject of today's session, or part of the subject of today's session, what do we even mean by AI? It's become a catch-all term for lots of different types of automation tools and technologies. I'm a big fan of the Information Commissioner's Office. Not many people are big fans of the ICO, but they've produced a really impressive range of materials on the subject. And I think the definition is one of the clearest. We define AI as an umbrella term for a range of algorithm-based technologies that solve complex tasks by carrying out functions that previously required human thinking. As with any other form of decision-making, those impacted by an AI-supported decision should be able to hold someone accountable for it. We're still in the midst of a massive hype cycle. Here are some of the breathless headlines from the last 6 months to a year about the wonderful transformational potential of AI. But those need to be counterbalanced with some of the less positive and very real problems and risks with AI adoption. It's riddled with bias, its results can be patchy, its environmental impact could be catastrophic. This image has been shared a lot on social media recently, just to highlight that AI can mean lots of different things in different contexts, and it's useful to be clear about what we're considering risks in terms of risks and ethical considerations. Everything's AI now. These are some examples of really exciting developments now in health and have come through AI and the use of health data. I'd particularly highlight the brain scans example. It's a project led by universities of Edinburgh and Dundee and involved researchers gaining access through the appropriate information governance processes to large numbers of brain scan images in order to develop tools that can help detect changes and predict risk. When we're considering how Research Data Scotland kind of tackles AI, we're thinking about in the context of research with sensitive data, and it's something that we've done a lot of kind of consideration of over the last year. It's not useful to make a blanket statement, so we've tried to break this down into the different things researchers might want to do. Maybe they use AI tools to help them refine their research questions or their code. That might be a question for their university or employers' AI policies, or for the ethical reviews they might want to have done of their research. As I mentioned, we only allow access to data in a safe setting called a trusted researcher environment. So we need to consider what is possible and allowable in that environment. The key thing is around outputs. For more traditional statistical outputs like tables or graphs, there are well-established methods called statistical disclosure controls to make sure those outputs can't reveal anything about an individual. However, this is harder to do with something like a machine learning model. It's difficult to test it fully, and there can be specific risks depending on how the model has been trained. This is a live area of work within our sector, and some of our partners are involved in projects to look into this further. Some of the foundational stuff that underpins the adoption of AI is the same stuff that should always be part of any technology or broader program. You need working technology and a sustainable infrastructure. You need good quality data and consistent standards. You need straightforward, well-understood policies. You need checks and balances in place. You need real use cases rather than technical fantasies. It's imperative for everyone that's involved in these processes to question if there's a genuine need being fulfilled. And a human in the loop. The one that differentiates AI is how to ensure there's still a human being involved to make sure checks and balances are properly in place and any decisions are being monitored and reviewed. Those are some useful resources, probably more useful if you are online and could link to these things. But with that, I'd like to say thank you very much.
Thank you very much. Thank you so much. That's fantastic. 3 fantastic talks, and very much looking forward to having some discussion now. So as I say, please do have your questions ready. If I can invite the panelists to join. I don't know why I'm moving that. Sorry. It's my control freak nature. I know, I don't know what I'm doing. I'll put it back. I will sit on a blue chair, yeah. Thank you. I keep moving it. There we go. Right. Thank you very much. So, I'm going to put the clicker down. I can't hold these on. There we go. Don't break that. Okay, so if I can first come to you both, Nick and Caroline. And Nick, I'll come to you first because you're closest to me. But what emerging challenges or opportunities do you see on the horizon in your area, and potentially in the area of the intersection between life and data sciences? If you see anything that's maybe not your area, but an area that's kind of untapped that we could be looking at.
I think one of the areas that's really, really important is, I think I'm going to go down to the funding landscape and the way in which when we apply for our funds as academics to do our research, AI is not assessed in the assessment process in any way, shape or form. Things like power calculations and clinical trials, they all have their statistics assessed, but AI is nothing. It just goes out to a person who might be familiar in the research area, let's say aging and frailty, but their expertise in AI might be completely negligible. So I think there needs to be more consideration of that in the funding landscape. I know it'll take more time and resource, but I think the previous speaker highlighted really, really well how we need to have a person in the loop, not just in making that, but also in assessing that. And how monies are handed out is really, really important to make sure that the science is underpinned by it. But I think having that assessment is really, really important. From a research perspective, I think some of the opportunities going forward are in linkage, are in how we link everything together. I give examples of the UK Biobank data. We've modelled, going back to the stuff I do, we've modelled frailty, we can do that. We've modelled the omics data, we can do that. What we're really interested now is the socioeconomic data and putting all of that onto that huge model. And I think that is a huge area that at the moment is very fractured, is very much all over the place. The social epidemiologists are coming at it from one angle, the clinical guys are coming at it from another angle. I, as a biochemist, are coming at it from sort of another angle. So I think that area is a really, really big area for opportunity going forward.
That's exactly what I was going to mention also, because I think the challenge and the opportunity is being better at bringing this different disciplines together. I know a little bit about infectious disease and models. Social scientists might know will definitely know much more about social determinants of health, and someone in a specific setting, clinicians in a specific hospital or a specific country will know how things work in that country. We need to be better at bringing all these disciplines, all these very important aspects together. We need to burst our bubbles and collaborate more.
Thank you. And do you think the ways in which to do that, where is the root of the problem? Is it around there being better funding for that kind of interdisciplinary work? Is it around the infrastructure that we need in Scotland and in the wider UK?
I think there is the funding. I think, like Nick mentioned, sometimes the funding might not be designed to be as multidisciplinary. But also, we need to be better at communication and translating our skills, our knowledge to different disciplines. That's something that I work on, and I try to not only training students or new professionals to be better interdisciplinary researchers and professionals, but also learning to communicate with different areas. So that's one of the projects that we had in our group a few months ago, communicating with artists, with NHS stakeholders, with researchers, data scientists. How can we find a common ground and learn how to communicate? Otherwise, we can be together but not speaking the same language. Mm-hmm. So that's a challenge to address also. That's why having this sort of situation, this sort of meetup and starting conversations is important.
I think we almost have to roll it back to education, don't we? We almost have to take it all the way back to the secondary school or even beforehand. As soon as somebody hits a computer, we almost have to have that mindset change, because that's what AI and all of these tools are doing. They're changing the mindset of the way that we communicate, of the way that we do science, of everything that we do. And I think that has to be reflected in the educational policies that we have going forward as well. Education is pretty siloed anyways. We all had our kind of, we had our maths double lecture, then we had our English, then we had our physics. And there's not, I don't know now if there's as much of that. It should be more integrated together. And I think that really needs to come. I think it's happening probably at a bit more at a master's level. I think it needs to almost drip feed down into the earlier years in our education.
And I think maybe to that and the later years, because I found that slide that you showed with what AI is, and then actually just all of the different things that people are describing when they say, and I'm guilty of that. I don't fully understand AI, and I have conversations with my relatives, and they're like, "Oh, is that AI?" I'm like, "No, it's not. It's just anything that's new in a technology." So I think there's educating. I think now, because it's also new and going to develop so quickly, educating all the way up, kind of to my level and above. I think so. But yes, I don't know, Hugh, if you've got any thoughts on that, around that education piece. Is there anything that you get involved with Research Data Scotland?
I think there's probably some really kind of similar points that Nick and Caroline have talked about. There's linkage in terms of data linkage being all important, but I think one of the big areas of opportunity at the moment feels that there's at least an acknowledgement that this stuff can't happen in silos, that the fragmentation that has been, I won't say allowed to prevail, it's just, it's sort of the natural order of things sometimes, but that things need to be better joined up. We had, the Goldacre review a few years ago, we've had the Sudlow review recently. We've got the UK government still talking quite vague terms about a national data library, but recognising that there needs to be the kind of infrastructure in place to support some of these initiatives, and part of the reason my organisation, Research Data Scotland, exists is that there is an understanding that we need to kind of not necessarily centralise, because it's not just about kind of pushing everything in one place, but have better networks. Caroline, you mentioned DASH, the trusted research environment that that Aberdeen University operates. There's now kind of a much more robust network of regional safe havens working with the National Safe Haven, who I kind of talk to on a daily basis, to think about how we do think through these issues together and think about solutions to problems that are more user-centred rather than kind of silo or business-centred.
Thank you. And I might take a moment to see if we do have any questions from the audience here with us today, and I think we definitely have a few maybe online, but yes, if you'd like to introduce yourself, that'd be great.
I'm Isabel from Isabel from Firefinch Software. I was just going to ask about the translation of the research that you're doing in the lab, and there's obviously huge amounts of things that could be translated out of the research that you're doing, and I think often it's been the job of the academic to try and find people to help you translate that. Do you think, it's maybe a loaded question, do you think that should be your job, or should it be somebody else helping to take that work out of the lab and support you there?
I'll take that one, shall I? Hi, Isabel, you're right. I think my view on that is, I think it should come from both ways. I think, you know, the academic should be open to the fact if you discover something of value to society, you should be up for getting it out there in the first instance. I think that comes as a responsibility from the funding. The vast majority of funding we get is from the UK, the public purse anyway. So I think absolutely you have that responsibility. That is a role now that you probably take on as an academic. Whether it's happened by stealth or not, it doesn't matter. I think that's your responsibility now to have that. I think as well from the other side, university-wise, we do have now universities have knowledge exchange centres and or other aspects to the university, other infrastructure within the university that encourages us to do things like that anyway, ultimately because there's a payday at the end of it. You know, there is money associated in inverted commas with that. To be sort of tongue-in-cheek, you know, if I invent a biomarker that can potentially predict if somebody's going to survive surgery or not and save millions of lives off the back of it, you bet your bottom dollar that's worth a lot of money at the end of the day. That's not why I'm in doing the job in any way, shape or form. I do it because I love the science and it's a cool thing to do. But I think you know, as academics, we absolutely have that responsibility. As academic institutions, fiscally, they have to have that responsibility to try and make the most of what all their brilliant brains are trying to make at the end of the day. And societally as well, I think we having sort of the way in which we can foster spin-outs, foster small companies as well to be able to do that. I mean, Strathclyde's got several companies that have come out in the last 4 or 5 years, kind of in some of the spaces I work as well, that have been really, really successful. But I think it takes that whole, again, it's that joined up thinking, it takes all of those players to be able to do that. It's difficult though, it takes so long and it's so expensive. But I think Scotland's quite good at it. I think we do have some of the universities, sorry, I'll rephrase that. Some of the universities are quite good at doing that. Some of them are very much not. And I think there are lessons to be learnt from all of the universities that we've got in Salsa and in the whole of Scotland to be able to make the most of it. So,
I was just going to absolutely agree. I think it's got to be a two-way process, and you know, just me coming here today and hearing both of you talk, you know, fills my brain with interesting thoughts about stuff that I just didn't know about. I think when our organisation was getting set up, when the kind of Scottish Government were doing the business case for it, there was a lot of user research done in terms of the communities, the research community, different academic institutions. Time and again, you hear people saying, I just don't know data's there, I don't know it exists, you know, the discovery tools aren't in place. There's an assumption that if you stick something on a website, people are just going to jump to it, find it somehow. So kind of the search and discovery tools need to be in place, kind of a consistent and coherent way about talking about research across disciplines needs to kind of be part of that as well, and just kind of promoting some of that join up in standards I think is a really important thing to make sure that we're sort of punching above our weight.
So just to add to what you both said, I think it's not 100% the researchers' responsibility to do everything, but I think it's the researchers' responsibility to involve key stakeholders from the start. So if it's about a project about with a health or a public health application, you should be including like a public patient, public involvement groups. You should be talking to key NHS stakeholders to think they should help design any projects, and they should help you to come up with the questions and so on. So like also when there is an impact, it would be much clearer what that means and what is the way to go. So I think, again, it comes back to the first question. We need to be better at interacting, communicating, and bringing different disciplines together.
Thank you. Any other questions from those here? Yes, and then we'll go to some online. Yeah.
Hello. Cheers. Hi, great talk from each of you. I was just wondering, from an academic standpoint, and I suppose for yourself, you'll have noticed that, because you've gone from maths, or physics, should I say, but into then using that, and I suppose that kind of bottom-up way of understanding kind of what's coming from the statistical analysis top-down is probably really valuable for you. But I was wondering for yourself, Nick, because I came from the chemistry with drug discovery at Strath. Do you think there's probably more space for less siloed undergraduate and postgraduate studies to be able to kind of have people almost with a more better intuition for like the math, statistics, and kind of everything that goes in to these sorts of things, so that it's not so siloed from like going from chemistry into maths? Because I know if you did it the other way around, you'd probably understand like a lot of things a lot more about maths and chemistry, if you get what I mean.
I think you get where you come from. I mean, I'm going to probably approach this from a bit left field. I think chemistry and the work I do, the bioanalysis stuff, is going to become automated. I think it's going to become, in the next 10, 50, 100, 200 years, it'll become automated. So it'll be a case of you'll have two people in a massive warehouse, generating all the data, right? We've got a huge, oh my goodness, what's his name? A huge professor at Glasgow University does all the computer stuff. What's his name again? All the Chemify stuff. Again, Lee Cronin, exactly. We've got Lee Cronin, who does all of this work with automating chemistry, and he's one of several groups worldwide that are beginning to get traction in that area. And if you can actually line that up, then how, not trying to put myself out of job here, but it's like, how much valid is it actually just having a degree in that area if it's all going to be automated? What's the point of doing it? What's the point of doing it? So then where does the need go? Well, the need goes to understanding what that chemistry can do, and what's the applied nature of what you get. When you do all these new molecules, what can you actually handle with it? How is it going to affect people in space and all the other stuff that you can do off the back of it? So I do agree, though. I think that there has to be again, it comes back to the educational bit we're kind of talking to before. There has to be a realignment. I think the AI, I'm not going to say just AI, but all of these tools that we're generating constantly, and the effect that they're having across the board now constantly, there has to be a redefinement in how we value our knowledge, how we generate that knowledge, how we educate that knowledge as well. And it goes across the board. It goes from cooking to chemistry to even to art, as you alluded to before as well, for good or for bad. I think all of that stuff has to subtly be, there has to be a big conversation educationally about that.
Thank you. I'm going to come down to you, Stephen. You got some questions online?
So I think this is for you, Nick, from Angelina, and she's asking, how did you quantify frailty in the UK Biobank?
Oh, so that's a very good question, quite a technical question. So there are several ways you can do it. The primary way, so there is a fantastic professor at Dalhousie University in Canada called Ken Rockwood, and Ken came up with this idea of a frailty index. And what a frailty index is, it's a concatenation of deficits model. What that really means in lay terms is, how many bad things have you got wrong with you, all right? That's what it really means. So, and basically you take 50 or 60 of these bad things, and it could be, how long does it take you to walk to the wall, to put your jacket on? Do you have cancer, a poor diet? Do you live in a poor SIMD area? All of these things get pulled into a model. Actually, that distribution you saw on my slide, that green right-skewed unimodal distribution that you saw, that was made from a frailty index. And there's lots of different ways in which you can make one, but it all stands the same, where you measure 50 or 60 things, and you divide the number of deficits by the total number of deficits in the model, and that gives you the value for the person. And if you're doing a population, you get that distribution. That's how you do it.
Thank you. Hopefully that answered the question for you online. Are there any other questions from here? Yes, hi.
John Kelly, currently Edinburgh University. I'm wondering, particularly with the healthcare data, so, and particularly in terms of providing longitudinal data, we've got a lot of information that's currently locked up in siloed sources, legacy sources, still in paper-based systems. And I was wondering what work there's been done to unlock that, which could give us some really useful information.
I'm going to go with you at the end there for that one. Absolutely.
I mean, I think there's a recognition that there is, yes, that exactly what you said, that in individual academic institutions, there's a lot of data locked up in government, in the public sector at large, all the different council bases. There is data, valuable data, which is not standardised, which is not currently accessible. Some of that's about, there's a slow drip feed kind of process. There's an awareness raising in the first instance. There's the getting the right standards communicated by which things can be properly tagged, properly assessed, properly evaluated and made available. I mean, I think that's where I would hope there's automation tools can come into play in terms of some of that data discovery, albeit with some of the caveats that I had in terms of my own presentation, where actually the kind of the digging around that's required to actually uncover kind of what's there can be automated, and actually kind of useful conversation with one of my colleagues in Scottish Government about some of the processes that they're looking at to improve some of the systems just to allow the surfacing of some of that siloed data. It's not easy, and it's, there's the whole, one of the kind of arguments for or against AI is the kind of the garbage in, garbage out. If you just accept what's there with no kind of basis or no standards in place, then what you're going to kind of come out with is not going to have the value that you might hope it would be. So there's a job to be done before that can just be sort of set out into the world and made available.
I think the standardization point is hugely important. I get that down at the molecular level of when we measure stuff. So it's like the genetic and the genetic stuff's quite good because the way in which you measure genes is actually quite standardized globally. So all of basically any sort of UKRI funded or NIH in the States funded, or at least it was until a few months ago, funded projects, all of the data would be generated on a similar platform, would be uploaded, and it's really comparable. You can make comparisons, you can pull it all together, and you can do some meta-analyses and that quite readily. The stuff I do with mass spectrometry, it's like the Wild West that is out there, simply because it's like the reason is because in like genetics, you've got 4 bits of chemistry, you've got 4 bases, your C's and G's and your A's and your T's, that's what you're measuring. I've got 250,000 different molecules to try to measure, so that becomes really, really challenging bioanalytically. And then when professorial ego comes into the equation, i.e. Professor X over in America says this is the best method, and Professor Y over in Scotland says this is the best method, then you've suddenly got 50 different best methods across the country. And we're still, from an omics perspective, the way that we measure stuff is quite lab dependent. And go back to your question of trying to put rubbish in, rubbish out. You need to standardize everything in the first instance, which is an issue. On the flip side, we do upload all of our data, and most labs do upload all of our data onto different websites. There are genetic, proteomics, metabolomics websites which all of our data should get uploaded onto. And those are quite large data sites now with terabytes and terabytes of data there. But it's just, as you said, what format are they in, how standardized are they, which becomes a problem. So it's probably a similar, different type of data, but similar problems that we actually face down when we're looking at the biomolecular stuff as well.
Thank you. And Caroline, I wonder, from just thinking around the work you did with COVID, and obviously it's something that everyone can relate to, but in terms of the data and the standardization of the data, there must have been so much data from so many different sources and different types of data, and I just wonder if you've got some insights on that.
Yeah, so even with this type of data, epidemiological, public health, there are different ways to aggregate data, to register data, especially about aggregation. Like someone decided to do a certain thing by week, aggregated the number of cases by week, by day, by the date of first symptom, by the date of recording that. So all this sort of things, or all this bias are underlying the data. We need to be aware of that when using it. So data is much more than just an Excel sheet, and much more than, you know, you need the metadata. We need to talk to people, the person that or talk or have information about who aggregated it, who collected. If you have a data dictionary, you need to know exactly what each of those fields mean. So yeah, there are no standards. My experience in LMIC, for example, different cities might have different standards. Imagine different countries, different states, different regions of the world. So, and then you try to compare all these things. So that's why, like a disease model, for example, my bias is this experience, right, won't work in a different place because the context is completely different. The healthcare system is completely different. What you measure, what you decide to record is going to be completely different. So data is much more than deciding what is the standard, but all this context to collect and record the data.
And do you think now, having gone through the global pandemic in this lifetime, the way the data is now, do you think the next time it'll be a bit better in terms of being able to work globally with similar data, or do you think still it will be as challenging because of the context?
Actually, one project that I was working on recently, commissioned by WHO in collaboration with University of Cornell. So the idea was to interview people at different regions of the WHO regions and produce a survey to collect data on like how their information systems for collecting public health data for infectious disease worked and what changed, what they learned during the pandemic. But it's just so difficult to get that information, and it's a huge challenge, and everything it's very difficult. The systems are completely difficult and completely different. And it's just like it's a political, financial challenge. Like we know what would be best, but like until you get all the approvals, all the bureaucracy to actually implement these things, I'm not sure it will be so much better so fast. I think we learned some of the challenges, the mistakes, but there's still a long way to go.
And I just add on that point that there was a Scottish Commission for pandemic preparedness, which reported at the end of last year. So there's a number of recommendations which are all about lessons learned when it comes to data access, when it comes to fleetness of foot, when it comes to where and how kind of the time frames that data's needed in, rather than the sort of, in some cases, months to years, needs to be down to days and weeks. So there are a number of recommendations that are sort of moving forward in terms of that from a kind of Scottish government sponsored level. So would hope that some of the kind of the key learnings that have come in from the what happened during COVID is now sort of in play and top of mind for some organisations, or for the relevant organisations.
Thank you. A bit of hope for everyone. Stephen, do you want me to come to another one online? Do we have another question? No? Oh, okay, lovely. Okay. Do we have any more questions from anyone here? Yes, hi.
Hello, my name's John Whitehill. I'm from Peak Scientific. I found this really interesting. I spend my life in the IT side of things, so actually hearing from you is just really good. But how on earth do you balance privacy in all of this? Because, and I'm sorry if it's a rabbit hole question, but working in corporate land for many years on things like privacy and cyber and all this kind of stuff, I know how difficult this is, but these data sets you're talking about are unbelievable. How on earth do you deal with all of that?
I mean, I can certainly sort of chip in my perspective. It's a fundamental in terms of everything that we're looking at doing as an organisation, the way in which so we talked a bit about trusted research environments. Both mine and Caroline's talk. The data that we're talking about is there's a process called the 5 safes that anyone who wants to access data for research purposes needs to kind of have gone through. Well, they need to have gone through, and the institution, as it kind of the institutions involved need to kind of, in terms of accreditation, in terms of safety checks. The data is, certain data we're looking at is non-personal. It's anonymised. In some cases, it will be pseudonymized, but in terms of removing those markers and keeping that, making that as many sensible, because there's got to be a balance of risk versus opportunity, but the right levels of safety and security are put in place. It's a live issue. I mean, again, speaking of the Information Commissioner's Office, they've published guidelines just this week about the anonymisation of data and when data counts as anonymised. So it is, it's something that obviously has to be brought in mind. But there are good protections in place and good processes to follow. Again, it's back to that. People need to know about those processes before they kind of start off in the process of making data available.
Yeah, thank you. From my experience, I haven't worked with data governance so much, but I have used, for example, individual level records, and but yeah, always anonymized. You don't have address, you don't have names, etc. And you need to, even for that kind of data, depending on where it comes from, you need to apply for it, submit a request. If we think about the safe havens, for example, the experience that I have, Dash is the Grampian Data Safe Haven. Their office is like 2 doors from mine, and if I want to use any data, I need to apply in advance, and there are like a bunch of checks they need to do. And if it's anonymized data, it might be okay, it might be aggregated, and then they might be able to produce that aggregated data for you to use. But if not, you can only access inside the safe haven, and so on. You cannot bring the data anywhere with you. So yeah, with healthcare data, there are like many more levels of checks that need to be done. Yeah.
I mean, from my side, I'm quite lucky at most of my stuff I get is anonymised anyway. Most of my stuff has had any individual characteristic, individual named characteristics taken out. But you have to be careful if there are things like, say we do go down the sort of socio-economic aspects, which we're starting to do in our lab. Obviously then you can begin to get things like postcodes, and you can begin to narrow down onto sort of individuals. Say there's a street that's got one postcode for one house, then you know exactly where they are. So you do have to be, it's not my area of expertise at all, that area, but obviously you do have to be informed, you do have to know what you can deal with. That's why I very much prefer it when I get in the lab, if we get stuff, it's completely anonymised. UK Biobank stuff's great because it is completely anonymised. It's anonymised to such a state that if you downloaded the same data set I downloaded, they'd have different names for the individuals, they'd have different codes for the individuals, so you can't even compare it. So there are methods in place there, but I think there is definitely still an ongoing educational, again, go back to education, educational piece that is in there that everybody has to be aware of, because it's really important, because you need people to contribute, you need the general public to contribute to these resources that we're generating, that we're building, and if you scare them off because you're being, fast and loose with like their data, as has happened previously, I mean, yeah, you're just going to, nobody's going to benefit from that.
Thank you. Yes, got another question from online.
So Deba Brata is asking, I think this is for you again, Nick. In frailty modelling, have you encountered situations where you rely on synthetic data in the absence of adequate data, and what potential challenges could arise in these circumstances?
Missing data, that's what we're talking about here, right? Missing data. Have you ever had data sets that have got missing data for frailty? The answer to that is sometimes yes. Often with like our biochemical chemical data, we'll get missing data. Say you analyse 1,000 samples and you're measuring 1,000 things, the chance that's 1,000,000 things. Chances are your instruments are going to miss a few things here or there. How we deal with that is imputation. There's various different ways in which we can impute that. But it depends what the project is. Sometimes we might just cut that person or that sample out of the equation because it's just not valid. I think there's a sort of almost like a range of severity of how you deal with such data sets. As I said, the severest is you just get rid of the sample that needs that sort of imputation done onto it. The other ones are more imputation based. There's KNNs, one of the ones that we use quite a lot of. But again, that's specific.
So it's synthetic data.
Synthetic data. So
Yes.
working on synthetic
Yes,
data.
rather than real data,
Do you
sorry.
know what? So right, here we go. I get where you come from. So sorry, I was viewing that from a completely different way. Synthetic data, do you know what? We don't necessarily deal with that much synthetic data because we generate a lot of our own. That's me being brutally honest. You know, we actually are, that's one of the main things we do is we generate physical, from physical samples, we will actually generate the data from first instance. So the answer to that is I don't have that much experience in it, in actually using it because we've got that much of our, I think, not blowing but we've got that much of our own to deal with in the first instance.
Okay, no problem. Thank you.
Well, I mean, in the context of modeling, it is useful, but mostly if you're developing framework in the more theoretical side of things to test your model, to try to predict parameters from that come from another model that you use to produce synthetic data. So it is useful, but usually when you are developing a modeling framework and testing it theoretically, but not in an applied health context.
Any other questions from the floor? Yes.
Sorry, this one came to my head, and I probably should have did this one first if I was going to do them. How much of a bottleneck is lab access to data and samples? So to make kind of a situational question here, like if you could get much easier access to data or even the analysis of a sample, let's just say there was a quick, easy pod at home that you could do it in, like a handheld side things. Is that attractive for people like yourself, Nick, and then obviously yourself, if you're trying to make more of a mathematical model? Would that be something of interest?
How much data is enough data? I suppose that's the question, isn't it? And the answer is probably never enough, right? I mean, I think if I was to, the question I'd answer to this is, how much money have I got, and do I have ethics, right? That's the first two questions that I've got. Because if I've got an infinite of the first and I don't need any of the second, I mean, what you would do is you'd take a blood sample of every person in the country or the planet and you would model that every day, right? And you track that and you would be able to do a hell of a lot with that data off the back of it. But obviously we can't do that for lots of the two reasons that I just mentioned, two reasons that I just mentioned there. I think it depends on how granular. I suppose what we're finding is with working with the UK Biobank, the reason that's such a fantastic resource is you can get really granular with your groups that you've made up. You know, we've got a rich tapestry of the Scottish population in this room here. We can split according to sex, gender, to ethnicity, to all of these things. Whereas if I was recruiting, say, that frailty trial that I recruited, predominantly they were white over 65 males that were in that study. Okay, the disease that we're looking at in the surgery sort of shepherded it that way. But I think being able to develop far larger data sets just allows us to be able to pick up the subphenotypes of disease that are out there. Because if you look at, say, breast cancer in the 1980s, there were two subphenotypes of breast cancer. There was, I think it was BRCA1, BRCA2, and non-BRCA. So it's 3 in total. It's 2 really, because it's BRCA or non-BRCA. Now there's about 16 subphenotypes of the disease that have each of their own different care pathways that we have. Different ways in which you do surgery, different drugs that we give them, different radiotherapy regimes that they'll have for those different ways in which we view breast cancer. And I think there's a lot of diseases that we've not picked apart like that yet. There's a lot of cancers and a lot of other things that we have not. I mean, for me, for aging, I think that, you know, there's 7 billion aging phenotypes out there, right? We've each got our own individual one. And it's like, how much data do you need to model that, right? And the answer is absolutely every single person in the room to be able to get your own one off the back of it. So I think the answer is yes. I'd love to be able to do that. Point me to it.
So thank you so much for all of the questions that have come from online and people in the room. I'm going to ask a final quick question and then we're going to do a wrap up. And that's really just, if we had this conversation again in 5 years' time, which I'm very happy to do, so I'll put a date in the diary, kind of what would success look like to you in terms of the impact of the work that you're doing now? And Hugh, maybe I'll come to you first.
I think success, it's a very unsexy answer, but infrastructure, it's about robust, secure, scalable infrastructure. It's having the basis, the basis that works, that's accessible, that is deriving results, so that some of the amazing research that's taking place is easier, is quicker, and is kind of providing long-term value in terms of outcomes. So yeah, so I think that would be it.
Thank you. No, it was lovely, succinct. I like it. Caroline, what about you?
I think I can think about like the analogous of infrastructure, but in the context of building networks and pipelines for us to use the data and people to be coming back to the educational training aspect of things, building networks, but not people from different bubbles talking about things that they don't understand, but actually being better at training people to communicate and work together. So I think success for me is building networks that can last longer and can actually lead to impactful results.
Thank you very much. Andy?
I mean, I think that, I suppose the way that we, I'm going to go on about the NHS here actually, the way in which the NHS deals with patient samples and the way in which we deal with that data, I think there's a lot we can learn from our colleagues over in America. I think the American Health Service is fantastic. It's the insurance service that's behind it that is not fantastic, okay? Once you're in the American Health Service, if you can afford it, you get all of the toys are thrown at you and you get the best treatment, some of the best treatment in the world. And what they tend to do is they will take a tumour and they will genetically sequence every single tumour that comes into the hospital. And I think, obviously we don't do that for cost reasons. It costs a lot of money off the back of it. And I think democratisation of that technology, ways in which we can get everything cheaper. And I think there's hope for that. I think when, if you went to the year 2000, I could sequence your human genome for the priceless sum of about $13.5 billion. I could do it now for about 100. Okay, and I think as that technology cost drops and the automation and all of that comes into it, I think there's very much scope to be able to, I just think, screen more and be able to do more of those screening technology. We've all probably been for a screen at some of us have been for a screen at some point within the NHS, and I think there's total scope to do a lot more of that. And obviously data and AI will play a huge role in the way that happens, and I'd just like to see that rolled out more, integrated more, and built into more ways in which we can, monitor people's health ongoing. I think I would like to see more of that.
So it just takes me, thank you for the panellists and for all of you, because you've made that a really easy session to chair. But just if we can thank our speakers again, thank you very much. Novel part of, I've never done this before, this is quite a cool idea, I might steal it for salsa things, but it's the kind of open mic part. If anyone here would like to do a kind of shout out, let us know about something you're doing from your company or your organisation that you'd like to speak about for a quick 30 seconds, I can give you the mic and you can do that. And it's a short time offer. Oh, we do, right, okay. David, I'll come to you first. If you could introduce yourself, please, and then just a quick...
Hi, good afternoon, everyone. So I'm David, and I'm here with my colleague Alicia from the University of Glasgow. So a lot of talk about education there. Sorry, guys. So a lot of talk about education there, which was fantastic. So I suppose what we are really keen to do is to connect with people who would be interested in working with the university. We've got a lot of new master's programmes that are coming out, MedTech Innovation to name but one. So if you're an organisation that's in that space that would like to work with us, helping our students about the next generation of people that are going into this field, Alicia and I would love to speak with you. So thank you.
Lovely. Thank you very much. Thank you. And then if you can stand up for me. Thank you. And then also there's a camera there just so you're aware.
Hello, cool. So I'm Adam. I am the founder of Scotia Biotech. We are looking to make essentially draw upon the experience of everyone who is speaking here, make kind of at the interface of data science, easy sample collection to make medical diagnostics much more accessible and easier to do. So rather than getting your samples sent away, if we can almost have a phlebotomist in any GP setting, just pop it on a strip and get some quantitative data out of that, and then progress that into improved, secure digital health records that can be useful for discovery through the cutting edge of academics, but also to improve treatment outcomes and things to make that much faster. So if anybody has resources, people that would be useful to put in touch, please let me know. It's starting. I'll have to say my cheesy tagline. We're taking the headaches out of concussion management, but we're looking to make it much further beyond that. And there's, I think, a lot of, talking about the kind of work that's being done here is like it's just the start, and I think it's a really exciting field for medicine at the minute.
Thank you. That sounds great. Yes, hi.
Hello, I'm going to move round because I'm really conscious that we're being filmed, and now I feel like I'm on Report in Scotland or something. My name's Marge. I am a little bit of an interloper, so Stephen very kindly and the team let me sneak in at the end, but it seemed like an incredible event, so I'm really sorry to have missed most of the discussions. I'm going to catch up with them. So I'm the community manager for Glasgow Codebase and TechScaler. The 3 pillars of TechScaler are community, mentorship and programmes. So we have a number of international programmes that you can do, like TechScaler Discovery, that you can do in your own time. Tomorrow is the launch of TechScaler Catalyst, which is an in-person and hybrid live delivery, 4 hours for 10 weeks for early stage startups who are either interested in investing and scaling, or halfway through the course it splits in half. If you're an earlier stage and you're looking to develop your MVP and you're developing software or applications, then you are welcome to apply. And that was my challenge, to get a lot of TechScaler information in a few sentences. But please approach me if you would like to find out more, if you're a founder, if you know a founder, if you're a student and your university or college supports spin-outs. Medtech and EdTech are the 2 out of our segmentation of 300 members in Glasgow and the west of Scotland, our 2 biggest categories. So we would really like to hear from more people in life sciences. Thank you very much.
Thank you very much, Marge. That's lovely. Okay, any more? Lovely. Right, don't go away yet. So I need to get the clicker. So we will have a little bit of time afterwards for a little bit more networking. So if there's anyone you've not had a chance to talk to, then you'll have a bit of time after this. I think we have the venue until 3:30. I am just going to take you through a couple of plugs. So there's a few things coming up. So the work we're doing in Salsa with the Data Lab community is a full month of events. So the next one coming up is on the 22nd of April. And this is going to be an online webinar, The Power of Data in Environmental Science from Genome to Ecosystems. And we have a couple of speakers from across our Salsa universities. And we'll have time for some Q&A and things like that. So that's at 11 o'clock on the 22nd of April. The one following that will be on the 30th of April, again online, which is going to be looking at molecules to population, so data-driven approaches to health research. And again, have 2 more fantastic speakers who will be there for that. So you can find out more on both the Data Lab website and also the Salsa website and register your interest for these. We then have DataFest 2025. So always such a brilliant event. It's going to be on the 12th to the 13th of May in the Assembly Rooms in Edinburgh. And again, lots of information will be on the Data Lab website, and that is currently open for registration, so get your tickets now. This one is, what we're doing this month is really building up to this funding call. So Salsa runs a number of different funding calls for researchers from across, as I say, our Salsa members, of which there are 13. And no doubt everyone here who's from a university is probably one of our members. So one of the funding calls that we run is called the Innovation Seed. We're delighted that the Data Lab is going to partner with us on this one this year, and hopefully looking forward as well. You can apply for between £5,000 and £15,000. You need to have an industry partner. It needs to be in life health sciences, and also has to have a strong data element to it. You do not have to be a life scientist to apply for it. And it really is some funding to be able to fund a translational project. We're going to open this in May. It is for independent researchers, so generally a PI. and you do have to have some match funding from industry, but it can be in kind. So today might be a nice event to be able to meet potentially somebody or someone through the Data Lab community. So I really would recommend joining the Data Lab community if you haven't already, because there are lots and lots and lots of industry on there and people who I think would be interested in connecting with you if you're an academic here in the room today. The last one for me to mention, I think, is Index 2025, which is a SALSA event, which is going to be running in October in Glasgow. It's a 2-day event. It's all focused on industry, academia engagement in kind of life sciences, health sciences. The first day is very much about R&D, about connecting with people around collaborative research. The second day, more of a skills recruitment fair and all kind of skills workshop looking at post-academic careers. And again, you can register your interest for that. Last one. This is really important because I think it has been a brilliant event today. I've really, really enjoyed it. Hopefully everyone online and here has as well. It really helps us to get feedback, to understand what's been useful and enable us to be able to continue to refine and make these kind of events the best they can be. So please do let us know what you think. And we'll be very careful with your data. And it's anonymous. There we go. So I think that is my last slide. So just finally, to really thank our speakers again, and thanks Stephen so much for all of his work and his team here today, Alan and Flo. So thank you very much, everybody. So yes, I think